All right, good morning. We are now going to start on our six-part tour through structural immunology. And structural immunology combines two concepts, two fields really. It's the biology of the immune system with the chemistry of proteins and cells. Where they meet is sort of structural immunology. It's where I've got my PhD in, and my postdoc work has been in it, my research has been in it in different ways. So I want to talk to you about where it all started. And to start with immunology, there's two places where you can go back to. And so let's go back to Sicily, 1882. There is a immunologist named Metchnikoff. At that point, he was a biologist, but he became the first immunologist. Now, I'm going to tell you a lot of these historical stories. These come from a really great article by Siddhartha Mukherjee, who's is written after the coronavirus. It was written in the end of last year, I believe. But it puts the coronavirus pandemic into the context of the entire fields of biology and history of science. And so we're going to start where Mukherjee starts. We're going to move all the way back to 1882. Now, the thing is, Metchnikoff's wife and kids had gone off to the circus, and he was messing around on the beach because he was a scientist. He would mess around with the animals, right? One of the things that he noticed that starfish larvae were transparent, and so you could see what was going on with the cells inside them. He actually had a eureka moment where he put some splinters into the larva, and he came back to the larva the next day, and he found out that not only had they had changed in a particular way. There were a bunch of cells that now surrounded the splinter. He called it the thick cushion layer. The thick cushion layer was made up of cells that he had seen doing eating other cells at other points. He called this phagocytosis, for the word for eating. I believe it's Greek. And they were called phagocytes because they were cells that ate other cells. And of course, when you see these cells eat other cells, the first thing you think is they're hungry somehow. They're eating them for food. They're eating them for energy. Well, he saw all these collected around the splinter, and he said to himself, well, they're not eating the splinter. They're not taking it in as energy. What they are doing is they are defending the starfish against it. So this is the kind of system that the starfish is involved in. He made some drawings, and these are some later drawings of but he used the transparent animals to be able to see that there was a cellular response to something that was foreign. There is a definition of self versus non-self. And when you look at the cellular response against non-self materials, you begin to start to have actual immunology. You are seeing the immune cells and therefore the immune system. He was a pretty good artist too. The thing that he noticed is because these starfish larvae didn't have blood, didn't have a brain, this system is going to be independent of both of those. And that's why I say he's the first immunologist. He's the first one to really isolate the system and to say this is different than these other complex systems that we have. They overlap, and the immune system definitely uses the blood and interfaces with the brain. But immune cells are not the same as red blood cells or as neurons. So in fact, these phagocytotic cells are one of the primary types of immune cells. They're the first ones that were discovered. And I just want to say, phagocytosis is one of the things we teach you in high school biology because it's so important. Here's a square of bacteria, and here is a neutrophil eating its way through. So you can see how originally they thought, oh, it's eating because it's hungry, but now we know it's an immune system cell. It's a neutrophil that is designed to eat for defense. It's not eating for energy. And this is just kind of a cool picture for what phagocytosis looks like. This is a very cool cell biology kind of phenomenon. But we can get down to the molecules that drive it, and then we can talk about the structures, the chemical structures that drive this immune cell, this neutrophil, which sort of has this automatic function to eat through invaders, to recognize them, and to destroy them by engulfing them. Here's what it's doing to bacteria. And so, because the immune cells work with a different system, they soon discovered that there's actually a whole network of vessels. It's like another road system that's in the body that will be independent of the blood and brain. It's called the lymph. And I can show you what the lymph is in several ways. I think the most vivid way to show you how the lymph works is to look at tattooed corpses. Okay, so this got dark pretty quickly, but there's a reason for this. A tattoo is an introduction of an ink that is not self. It is a long-lived molecule that is colored, that is subcutaneous, and it lasts for the rest of your life. As you know, if you've been thinking about getting a tattoo, that thing is very hard to remove. It takes lasers and stuff like that. So the thing about this is a foreign substance in the body. The immune system actually will sample it. And what the immune system will do is it will sample it, take it through the lymph to lymph nodes, where it will sort of put it through the test to see, is this me or not me? Well, it's not me. Okay, is this dangerous or not? Should I learn to tolerate it, or should I freak out? And that's what the lymph nodes are there to do. So it's important to see these as sort of central locations. And there are lymphocytes that move through the lymph. Those are the cells that we're talking about. So here you have the idea. The tattoo is introducing a foreign substance, and some of the tattoo will just passively diffuse through the body, but there are active cells, and you see that they look like little trees. There are dendritic cells, are one of the types of cells. Their job is to scour the body, look for stuff, and bring it back to the lymph node. And they do that with the little tattoo particles. Because tattoo particles are colored in the skin, they are also colored in the lymph nodes. And that's what you can see. When you biopsy lymph nodes, you can actually see the different tattoo colors that have been brought to it. You can identify it back down to the exact structure of the pigments. We have this azopigment that has colored the skin green, and you can find it also in the lymph node. There are different copper pigments that you can find, and when you look at the lymph node, you see that dark spot in the lymph node. That is the foreign body having been brought back to it. So the lymph node vessels allow the cells, the lymphocytes, to be able to carry these back for sampling. In the case of tattoos, it's not a problem. In the case of bacterial infection, maybe they'll decide this is something we need to mount an immune response against. Then a whole bunch of different cells get together and go after the bacteria. So this is really the picture of what it looks like. The lymph nodes are scattered throughout the body, and there are lymph vessels that connect the lymph nodes. It's kind of like another set of roads. The blood and the heart, main roads of the body that carry oxygen and all sorts of other things. But the lymph is a interconnected set of roads, and the important thing that it connects, so the other part of the immune system that's important is, where do the immune cells come from? In our case, they come mostly from the bone marrow, and that is where also the red blood cells come from. So you can see that the white blood cells, which are involved in the immune system, come from the same place as the red blood cells. They come from the bone marrow. The only other thing that you'll see is you'll see a couple other things that are involved with the immune system: the spleen, the tonsils, and the thymus. The thymus is actually very important for a particular type of immune cell, and we'll get to that in a couple of parts. Tonsils and spleen have less central roles in the immune system, but they definitely have roles to play. They're part of the whole immune response, which is why the tonsils sometimes get swollen with immune cells and fluid and things like that. So we're going to be talking about the thymus eventually, and we're going to also be talking about bone marrow and lymph nodes. So why I was a little bit wondering whether Metchnikoff is truly the first immunologist, because you might know about vaccines, and vaccines definitely predate 1882. In fact, vaccines go all the way back. One of the earliest ones we can find is in China in 1500. Smallpox was a devastating disease, and it actually had several versions that infected different species, including domestic animals: horsepox, cowpox. So smallpox was such a bad thing, but people began to realize if you were able to survive smallpox once, you never got it again. And so medical healers in China made this observation, and they inferred that there is some kind of exposure you can do. So they tried to thread the needle. They tried to say to themselves, let's give somebody enough smallpox to educate their immune system, enough smallpox so we can recognize it, but not so much that it actually causes disease. And you actually can do this. This is kind of gross, but they ground smallpox scabs into a powder, and they blew it into a child's nose with a long pipe. And many times, this would actually give the child maybe a mild case of smallpox that they would recover from, and then they would never get it again. It's kind of like a chickenpox party that we talk about and things like that. And so this idea of getting just the right amount of immunity is an old idea. It's also very dangerous, because then you have an intervention that you're deliberately making the person sick. So you have to be very careful with it. Immunology is really a case of educating your immune system with less and less chances of you getting sick yourself. Educating it well on the dangerous thing without actually giving the dangerous thing to you. This is a case of doing that by having a small dose of the dangerous thing. And it's only the very first step. It doesn't work very well, and many people do get sick, and some people die from the intervention. So this same idea was carried out in England by Edward Jenner, and he took cowpox and he gave it to an 8-year-old named James Phipps. Informed consent was not there. There were all sorts of problems with how he did this. Thankfully, it actually worked, because 2 months later he actually gave live smallpox to an 8-year-old, and the Phipps did not get sick from the smallpox, and he survived, and so Jenner knew that his idea worked. This is why the word vaccinate comes from this practice. It comes from the fact that you can take a cowpox, and it's similar enough to smallpox, human smallpox, that the cowpox will educate the immune system to reject and react against the human smallpox, so that you have the memory of it without ever having actually technically encountered it. Cowpox is less deadly, and that's one of the reasons why this whole thing works. It's less deadly for humans. This is also why the word vaccinate says vacca in it. Vacca is the Latin for cow, and so you're giving cowpox, you are vaccinating. But you see that this is still very dangerous, and you have to persuade people that you know what you're doing. And you didn't always know what you were doing back then. Apparently Jenner was confident, but that led to some medical ethics violations. And so from the beginning, vaccines have been a very tricky area to navigate, okay? But also very useful. I mean, not getting smallpox is a devastating disease, and so it's a very good thing to be able to avoid smallpox. But people were anxious about it. They're anxious about deliberately giving yourself cowpox to avoid getting smallpox. Here is a cartoon that shows a doctor giving cowpox to people as a vaccination. And you see all these people, they're turning into cows, on the other hand. That was the fear, and this is an expression of the person's fear, which is an expression of a very real anxiety that maybe we don't have it right, or maybe my immune system is different from most people's, and maybe I don't react, and maybe this will actually be bad for me. This anxiety is very old, and we are still dealing with it. And I have to say, as a scientist, I cannot eliminate it. There's always going to be some risk with vaccination, but we can minimize that risk. Okay, so a little bit more as we move through history. Vaccines defend you against bacteria. They also defend you against toxins. Certain toxins will activate the immune system. And they found out that, for example, if you had a tetanus, means that you have the diphtheria toxin in the blood. And tetanus affects animals as well as humans. So they took blood serum from one animal and they gave it to another animal. And they found out that it would protect that animal against tetanus, against the diphtheria toxin. So they concluded from this that the blood was carrying something that was an antitoxin molecules. I believe they could rule out that it was cellular based. I believe that they knew that it was a molecule, a body as they called it, as opposed to a living cell. Okay? So Paul Ehrlich was the immunologist who called them anti-corpers, because I believe that's what it is in German. But they are, corper means body, and so in English that term becomes antibodies. Now they are antitoxin bodies, is sort of abbreviating out the toxin in there. But of course now we've used the term frequently and without even thinking about where it came from. Eventually they found that antibodies, the chemicals that were the antibodies, proteins, were made by a particular kind of B cell that was had matured into what's called a plasma cell. A B cell turns into a plasma cell and makes a bunch of antibodies when there is an infection that you remember. Okay? And you can take those antibodies and you can actually use them as a therapeutic. And they're even talking about, it's one of the therapeutics we have for COVID, is to take monoclonal antibodies, a particular kind and purified form of antibody, and to give it to the patient. They are proteins, and so you can do a lot of things with them. You don't have to worry about keeping cells alive. You just give them the proteins. They're a lot more stable. So the thing is that they chased down what made the antibodies. They found out that these cells that they knew were in the white blood cells, called B cells, were the ones that actually made the antibodies when they grew up. They matured into plasma cells. They were activated. And so they're looking a lot at where these B cells came from, and they found out they came from the bursa in the chicken. Now, I believe the term B cell actually technically predates bursa, and so B is not for bursa, but you can associate that if it helps you remember. But humans don't have a bursa. So they looked around for the equivalent of the bursa in humans. They found out that the bone marrow is what makes the B cells. I believe this is a coincidence twice over, that bone marrow starts with B, but there we have it. You can remember it that way. It doesn't matter if it historically technically happened that way, if it helps you remember it. The bone marrow is where the B cells come from. It's also where red blood cells come from. So both components of the blood are produced in the same place. Now actually, if you look down, there's actually, so the B cells come from the bone marrow. And then they found out that there's other types of cells. More on this later, but the T cells also come from the bone marrow. But T cells have a little bit of education they have to go through. They go to the thymus and they literally become educated about their job of discriminating self from non-self. That's going to come much later in the story, because you notice that this is based, T cells work based on the whole cell working together. T cells are more complex than B cells that just make antibodies. So we're going to stick with the antibodies for now, but don't worry, we'll get to the thymus eventually, probably next week. The B cells and T cells both sort of migrate around, and they migrate around the spleen and lymph nodes. The lymph nodes are sort of headquarters where the B cells and T cells figure out whether they should attack or not, whether they should make antibodies, whether the T cell should modulate immunity. And the B cells, the other thing I want to mention, the B cells have receptors on them that look just like antibodies. It's like each B cell has these antibody-looking receptors, but they're bound to the B cell membrane. And so it's a very clear mechanism. The B cell gets activated. It has the antibody on its surface, sort of as a receptor. And then it makes more of that antibody just as a free-floating protein that can go around and that can bind the antigen. Okay, the antigen generates antibodies. And those are the important terms that you can talk about. There's little plasma cells down at the bottom. You can see that they definitely look different. The cells look different when they become activated. So, at the end of this part, I would like you to go ahead. This is just the first part of 6 that are spread out over 2 weeks. But this is near the end of part 1. And so I want you to see a movie of how in the world does a B cell know to respond, to get activated, and why does the system work that way? Well, the system works that way because you have millions of different B cells, each of which has a single antibody receptor on it. In fact, this solved a long-standing problem in immunology, which was, how do you make antibodies to antigens that you've never seen before? Okay? There were 2 ideas that maybe you had millions upon millions of antibodies. That didn't make sense. That's a lot of antibodies to make. and how do they generate themselves? The other idea is maybe the antibodies mold themselves around the antigen somehow. But how does that recorded? How does that work with the cell, with the protein making like we know? It didn't make sense. And it was solved by Frank Macfarlane Burnett. His mother was a Macfarlane, so we might be related somewhere in there. But he was a Melbourne scientist, Australian Journal of Sciences, where he published this obscure idea. We solved this problem, where he said, what if the B cells are what make the antibodies, and each B cell is an individual antibody machine. Therefore, you can activate the cell, and it can make more antibodies all at once. It makes more clones of itself, and therefore this is called clonal selection. So I want you to watch this movie. It's about a 5-minute movie, and it shows you exactly how clonal selection works to proliferate B cells into plasma cells, into bunches of antibodies. The key thing is the idea that each B cell has its own particular antibody that it makes. And so out of your millions of B cells, one of them is going to be able to bind at least a little bit, and then the B cells can evolve through a process called affinity maturation, by which they increase their affinity. But there's more of those. We're just talking about the cells right now. So this is what the one looks like. I believe it's on Canvas or on the playlist. I'll put it. And if this reminds you of Star Wars, Attack of the Clones, that is definitely an illusion that immunologists have thought of before, ever since that movie came out. These are literally clone wars going on in your body. So that leads to lots of scientific puns and lots of explanations for what immunity is like. So for biochemistry students, I have also put up these 4-page comics. Each of the pages is a different comic, and they're about how immunity in general works and how RNA vaccines work. This is another angle. I want you to look at it. I want you to think about it, but we'll be talking about these concepts as we go on. But it also will tell you more about something I don't have time to get into. How do the mRNA vaccines that we have against COVID, how do they work? These are really nice explainers for those. So that gives you some more things to look at. That ends part 1. And then once you look at those and come back, we will get into part 2, where I'll tell you about the structure of antibodies, how they work as proteins.